Welcome to NerdyGenes. In this video, we are going to see about restriction enzymes, which is also called as restriction endonuclease. To be precise, endonucleases cleave phosphodiester bonds in nucleic acid. On the other hand, exonucleases removes nucleotides from the ends of DNA or RNA. In short, endonuclease cuts DNA in the middle and the exonuclease cuts the DNA at the ends. Restriction enzymes is also called as molecular scissors as it helps us to cut desired DNA fragments. Let's see a short overview on how it works. First, it scans the entire DNA to find its specific site to cut. Each restriction enzyme has its distinct set of sequence on which it can act, and that is called restriction site or recognition site. And after spotting its site, it makes a cut. The restriction sites are usually palindromic sequences. In DNA, palindromic sequences are those that are same when read forward and backward on complementary strands. For example, let's take EcoR1 restriction enzyme. Its restriction site is GAA-TTC and its complementary sequence is just the reverse version of it, and that is CTT-AAG. When read from 5' to 3' on both the strands, it's GAA-TTC. Everything is fine. But where do we get these restriction enzymes? We get them from bacteria and also archaea. But bacteria is the primary source of restriction enzymes. These bacteria produce restriction enzyme to protect themselves from bacteriophages, and that is viruses. These restriction enzymes cleave viral DNA and act as a defensive agent. And we may have a doubt like, how come bacteria's DNA is not cleaved by restriction enzymes? And that is because the restriction site in bacterial DNA are methylated, which makes difficult for restriction enzymes to cleave it. By just seeing the name, we can identify the source of the enzyme. For example, EcoR1. Eco means it is from E. coli. R denotes its strain. 1 denotes that it is the first restriction enzyme to be discovered from R strain of E. coli. Let's see about the types of restriction enzymes, and this classification is based on its function. There are 3 types, type 1, type 2 and type 3. Type 1 and type 3 restriction enzymes cleave the DNA at a place that is far away from their recognition site. A little difference between them is that type 1 cuts DNA at several thousand base pairs away from its restriction site, but type 3 cleaves at a site that is 20 to 30 base pairs away from its restriction site. Example for type 1 is EcoK1 and for type 3 is EcoP151. Then moving on to type 2. Type 2 restriction enzymes are those that cleave exactly at its restriction site and holds a number of biotechnological applications. EcoR1, HIN3, BAMH1 are some of the examples of type 2 restriction enzymes. Based on mode of action, that is cutting fashion of restriction enzyme, they are classified into 2. Blunt end cutters and cohesive end cutters. Blunt end cutters cut both DNA strands at the same position within their recognition sequence, resulting in blunt ends where there are no overhanging nucleotides. For example, let's see about SMA1. It recognizes the sequence CCC, GGG, and it cuts between C and G and also produces blunt ends. And the next type is sticky end cutters, also known as cohesive end cutters. These enzymes cut DNA strands at staggered position within or near their recognition sequence, and that results in overhanging ends known as sticky ends. These overhangs can be either 5' or 3' overhangs depending on where the enzyme cuts. For example, EcoR1 leads to an overhang in 5' end and KPN1 cuts between C and C and produces an overhang in 3' end. Let's have a glimpse on isozymes. Isozymes are those that have same restriction site. They are of 2 types, isosisomers and neosisomers. Isosisomers have same restriction site and also cuts in the same fashion. For example, SPH1 and BBU1. They both have same restriction site, that is CGTA-CG, and both of them cut between C and G, producing sticky ends with overhanging in 3' end. And neosisomers have same restriction site but they differ on cutting patterns. For example, TAI1 and MAE2. They both have same restriction site, that is ACGT, but MAE2 cuts between A and C, producing sticky ends at 5' end. On the other hand, TI1 produces blunt ends. Let us say that we have a DNA of interest that needs to be cloned. So to isolate that DNA of interest, we use the restriction enzyme. Let us say EcoR1. Now we need to add it to the vector, but the vector doesn't have EcoR1 restriction sequence in it. So to overcome this issue, we have linkers and adapters. Linkers are blunt on both the ends and they introduce new restriction sites into the DNA, which is then digested with the needed restriction enzyme. And whereas the adapters have a blunt end on one side and sticky end on the other side, it converts blunt ends to the sticky ends when added to it. Restriction enzymes work efficiently and effectively when they are provided with optimal conditions, like pH lesser than 8, glycerol concentration lesser than 5%, high ionic strength, etc. But when these ideal conditions are not provided, they make non-specific cuts apart from their restriction sites, and this activity is known as SAR activity. Restriction enzymes have a wide range of applications in molecular biology, genetics, and biotechnology. Here are some of their key applications. In gene cloning, restriction enzymes are used to cut DNA at specific sites to create fragments that can be inserted into plasmid vectors. These plasmids can then be introduced into bacteria to replicate the DNA, a process called gene cloning. In genetic engineering, restriction enzymes allow scientists to manipulate DNA by cutting and pasting genes from one organism into another. This technology is fundamental to create GMOs, that is, genetically modified organisms. In molecular diagnostics, restriction enzymes are used in techniques like RFLP analysis to detect genetic variation. This method can be applied to diagnose genetic disorders, identify individuals in forensic analysis, or even determine paternity. In gene mapping, by cutting DNA with different restriction enzymes and analyzing the resulting fragment sizes, scientists can map the location of genes on a chromosome. This technique was crucial in early genome mapping projects. In recombinant DNA technology, restriction enzymes are used to create recombinant DNA molecules by cutting DNA from two different sources and ligating them together. This is fundamental in the production of insulin, growth hormones, and other therapeutic proteins. In southern blotting, restriction enzymes are used to fragment DNA before separating it via gel electrophoresis. This method helps in detecting specific DNA sequence within a complex mixture. These applications make restriction enzymes invaluable tool in research, medicine, and also biotechnology. Questions on this topic will be discussed in another video. If you have any doubt, please comment below, and thanks for watching.